biomolecules. Now this is one chapter which comes after amines like amines and alkyl halides. If you remember earlier, some of us who were there for a couple of years know that the organic chapter, the part 2, alkyl halides, aryl halides, alcohols, phenols, amines and biomolecules carried the same weightage, 4 marks. Earlier we used to have chapter wise weightage, if you remember. Now they don't give the chapter wise weightage. Only aldehydes, ketones and carboxylic acid had the highest, second highest weightage after P block. P block had the highest weightage. Correct me if I'm wrong. That was how the weightage was earlier. So what I'm trying to tell you here is the chapter biomolecules is as important as alkyl halides, aryl halides, alcohols, phenols, and amines chapter. Many places I have heard, I have seen where the chapter biomolecule is converted to a seminar. Not wrong, a more flipped classroom kind of session where peer group learning happens, because peer group learning is another very important methodology. We can have that provided children are empowered with knowledge. Without empowering children and asking them to do a seminar, especially children who do not have the basis of biology, because in 11th standard they get to choose whether they can leave out biology if they want. Children who go in for CS, I don't know if your schools have, do you have any option leaving out biology?
Yes, ma'am. Is that
CS is there, ma'am.
CS is there, yes. So when we have a classroom full of CS students and expect them to do justice, probably they'll work over and hard and then come and then do the presentation. But that should be done only after empowering the students with the knowledge, the way of presentation. So this chapter is got equal weightage as the 3 other chapters I mentioned. Only aldehydes, ketones, carboxylic acid carried earlier a little more weightage. Now I think they overall balance that by giving certain marks for organic chemistry. Next year, 2023 board exam, we are going to have all the 10 units, though they have left out some very, very important, I understand, very useful units. I am still wondering why P block was left off.
Yes, ma'am.
It's a challenging lesson, but learning challenging chapters help children evolve to handling challenging situations. I always say on the lighter side, when you are able to handle chemistry, you're able to handle the chemistry teacher, you're able to handle many difficult situations in life. Because chemistry subject itself transacting in a friendly note with a anecdote in between becomes a challenge. I don't know. I'm not able to come up with anecdotes, funny ones in the class to make it a little light. We can't. At certain topics, we can't afford to make it light, because when it goes in light to students, they think it's like, okay, not so important. So that intensity needs to be felt within us for children to feel that intensity. So chemistry is always their third favorite, or maybe sometimes the fourth favorite also, unless they have a strong bonding with the teacher. It becomes their not so favorite subject. Going on to this chapter, biomolecules, like I said, I handle it the same way, give it the same importance that I give tell children also the same. So just sharing the PPT here. You're able to see the screen?
Yes, ma'am. Yes.
Yes, ma'am.
Okay. So learning outcomes, as usual, we'll take up the learning outcomes. Students will be able to classify carbohydrates and appreciate the structures. Students will be able to draw the open chain and the cyclic structure of glucose. This comes only after children know how to deduce the structure. This deducing structure of glucose, they have not asked till now. Have they? Have I missed any time where they have asked us? They never asked to deduce. They say reaction of glucose with maybe concentrated nitric acid. What structural evidence do you get from that? Treating with HI, what structural evidence? What is the product and what evidence? Yes. So that clarity step by step, as we studied in our organic chemistry in our masters, where structural deducing structure, elucidation was, they're not asked in the board exam till now. But then appreciating, children should be able to appreciate the fact that these structures are arrived only after chemical tests, rigorous chemical tests. So, and at the end of the test, children should be able to, after the series of tests, children should be able to draw the open chain and cyclic structure. Yes, cyclic structure has been asked in the board exam, but then it requires, again, one observation that I have made through as a teacher, I have noticed that children, we find it difficult, but they have the spatial arrangement, they understand it better, faster also. And when it comes from their peer group, who's more strongly prepared, it is also done very well. But then a word of caution here, not give it to students who are not given or have enough experience. They could sabotage the whole effort. They could put up things on the board which they don't understand and also can confuse. So be very careful when you go on to ask children to draw the structures unless they are thorough with it. Students will be able to identify disaccharides and polysaccharides. Please stop me in between if we need to discuss anything in detail. And I am given to understand nowhere it is given that structure of lactose, maltose and sucrose will not be asked. It is not said, but it is not asked. It is unwritten kind of. Am I correct? Or is there any cases where those questions have been asked? They probably asked product of hydrolysis of sucrose, maltose, lactose. They have not asked the structure. But then it is it's a, you give them the structure, the cyclic structure, the pyranose structure for them, and then give them the glycosidic linkage and ask them to draw. They come up with very well. Just have to give it to them in their rough notes on their class works, ask them to draw, so that they'll remember what are the products of hydrolysis of the disaccharides. So that is not just identifying them, but also identifying them with their monomers become very, very important. Students will be able to appreciate the role of amino acids in living organisms and its classifications. Again, there was 1 year board exam, I remember. How are amino acids classified based on their how are they? The answer I remember, but I don't remember the question very well. The answer is acidic, basic, and neutral. Remember the question, Lakshmi ma'am? What was the question?
I think they had given no, they had asked
basic and neutral. I think something like that.
Yes, the expected answer was acidic, basic, and neutral, isn't it, ma'am? But they're not. They phrased it in a way that they're not asking about essential and non-essential. They were not asking that.
Yeah.
They
With
said
an example, with an example.
yeah, a paragraph or something they said, and then they said, how would you classify amino acids? So we normally go with essential and non-essential, and we don't give much of importance to acidic, basic, and neutral amino acids normally. But then that is also one way to classify amino acids. Another very important aspect as we are going on, we'll be discussing that amino acids are like ionic solids. That part is also a very important give reason question because of the formation of Zwitter ion, the salt-like properties they have. And alpha amino acids only can do it. So alpha halo acid and alpha amino acids, comparing that. So by comparison also, the child will be able to appreciate the difference between halo acids and alpha halo acids and alpha amino acids. And children will be able to relate to the connection between amino acids and then proteins. Amino acids and proteins organically evolve through proteins. Again, like I always tell children, very rarely will you be asked questions on like define carbohydrates, and they can't define carbohydrates as energy giving foods or bodybuilding foods, bodybuilding food for proteins. They need to give the chemical definition. Optically active polyhydroxyaldehydes or ketones, or those which upon hydrolysis give optically active. So that definition which is there in the first part of the textbook. Similarly for proteins, polypeptide chains containing more than 100 amino acids with molecular mass 10,000 U exception is there. With the exception, if they remember, so these definitions come integral when they are able to understand what these are. And general trend is CS students give this lesson a stepmotherly treatment. Correct me if I am wrong.
Yes, ma'am.
Computer science students feel that I gave up biology
Yes,
because
ma'am.
I don't like it, and they are
Yes,
making me
correct.
study this. So I'm not read. This chapter,
Yes.
if handled properly, they can score very good marks. So that we can make it happen if we kind of give them the right way to approach it, right way to handle it. They are scared of the structure. They're scared of the a lot of content in biology. Biology is not very tough, but then there's a lot of volume to it, and that volume is what scares students. So if we can give the content as the form of a concept map or a mind map, every chapter can be done. We can mind map a chapter, but chapters like this, when we mind map it, children find it very comfortable, because this particular chapter is bordering by a lot of biology also. The next is students will be able to appreciate the role of enzymes and hormones in biochemical reactions of an organism. Hormones are there, ma'am. Are they still for evaluation? Hormones, I understand, are not for evaluation,
not
but
for
then it's
evaluation.
there in the textbook. It is there that they've added as an appendix as a last part, additional page. They've not integrated it with biomolecules yet. At least the older edition has it at the last. But then just knowing that it is not going to hurt the child. I guess we should be able to, students will be able to understand the importance of vitamins in the function. Yeah, this is very, very important. I think vitamins also is removed, isn't it, ma'am? Last year, at
Last
least
time,
it was-
ma'am.
Last year it was
Last
removed,
year.
ma'am. This year it is there.
it's there. Okay. Okay. Students, again, the role of nucleic acids, appreciate the role of nucleic acids. The RNA, and this is basically the overall learning outcomes that we expect from students. Key points, the key headings, carbohydrates or what are there in the textbook. Methodology, we can help with recall. Flipped classroom is normally done in the chapter. Visualization, use of blackboard, very, very important. Differentiation, peer instruction. Like I said, peer instruction helps. When we have a class of biology and CS students both together, the biology students come handy at this time and they can be used. And drill project also helps them kind of, gather information and come up with data. And with the data collection, we can analyze. Going on to the content itself, just thought we could discuss a few things because this particular chapter, not necessary they have to learn by heart the structures, except for glucose, alpha D glucose. One small way to help them remember is always help them remember with the open chain structure of glucose. This is sample paper question says, what does alpha D plus stand in? The name alpha D plus glucose in the pyranose structure. So how do we help them understand that? What does the D stand for? Anybody?
Dextrorotatory.
No, ma'am. The capital D doesn't stand for dextrorotatory, ma'am. The positive sign stands for
Capital D.
dextrorotatory. Capital D. Capital D.
Sorry.
The capital D represents the configuration of the molecule, ma'am. Maybe it
So it's-
represents the fifth carbon contained
Correct, sir.
OH group at the right side, ma'am.
Correct, sir. Very good, sir. Very correct. It stands for the configuration. It stands, the D stands for the presence of OH group on the right hand side of the last chiral carbon. Am I right, sir?
Yes, ma'am.
Last
Yes,
chiral
last
carbon.
chiral carbon. Yes,
Last chiral carbon or the fifth carbon.
fifth carbon. Yes.
Carbon, because we start numbering from 1 of the aldehyde carbon. So we
Yes,
can
ma'am.
either say OH group on the right side of the last chiral carbon, or OH group on the fifth carbon of the glucose structure. So what does the alpha stand for? What does the alpha stand for? Alpha stands for the presence of OH on the right hand side of the cyclic structure of the C1 carbon of the cyclic structure of glucose. When we have the hemiacetal form, we have the OH on the right hand side of the C1 carbon of the cyclic structure of, that's what alpha stands for. That's where we say the proof, that's one of the proof for the fact that glucose also enjoys a cyclic structure along with an open chain structure. The presence of two crystalline forms, the alpha and the beta form. It's a very important. What are the evidences to show that glucose not only exists in the open chain form, it also exists as a cyclic form? What are the evidences? Anybody remembers? It's a different paragraph itself in our textbook. What are the evidences to show that glucose exists in a cyclic structure also, not just have an open chain structure? Anybody with any thoughts on this?
Shifts base.
It does not answer Schiff's base, correct, ma'am?
Yes, ma'am.
There are 3 points given in our textbook.
Yes, ma'am. Yes.
It does not form hydrogen sulfide with the
It
sodium.
does not reaction with sodium bisulfite. Addition product with sodium bisulfite, correct?
The pentaacetate form of glucose.
The pentabustate does not react with hydroxylamine, right, ma'am? The third point given in the textbook is the existence of two forms, the alpha and the beta form of crystalline glucose. Here, anybody would like to throw light on what are anomers? And in carbohydrates, the whole discussion is about glucose. What are anomers? Anybody would like to talk about it?
Which is forms as a chain from first carbon to fifth carbon, ma'am.
Sir?
It forms as a cyclic structures.
From the first carbon to the fifth
Differentiation
carbon, sir,
to
the cyclic
first carbon.
structure
Yes,
with
ma'am.
the OH on the right hand side and left hand side. OH on the right hand side is the alpha form, OH on the left hand side is the beta form. These two are called the anomers, and the aldehyde carbon is the anomeric carbon. Am I right?
Yes, ma'am.
The anomeric carbon is the after cyclization becomes the anomeric carbon, because that's the configuration of that carbon differentiates between the alpha and the beta form, and they have the melting points also very different. And that is what the third evidence for the fact that glucose also exists in the cyclic form. Again, encouraging students. How would you encourage students to try out the structures of glucose? Apart from giving them those reactions with HI, let's quickly recall what are the reactions. Reactions with HI, we have reaction with bromine water, reaction with nitric
Oxidation.
acid, reaction with hydroxylamine and HCN. To talk about the presence of aldehyde group. What is that reaction which helps us identify that there is a primary alcohol? Which reaction of glucose helps us identify the presence of primary alcohol?
On oxidation with
On oxidation, ma'am? On oxidation with what, ma'am?
bromine.
Oxidation with bromine gives us gluconic acid. Am I right, ma'am?
Yes, ma'am.
That's a mild oxidizing agent. So mild oxidizing agent converts aldehyde to carboxylic acid, which is gluconic
Okay.
acid.
Yes, ma'am. Yes. Saccharic acid we will get, no?
Saccharic acid with nitric acid, ma'am. With
Yes, yes,
concentrated nitric acid, when we get saccharic acid, we say this is apart from the
Aldehyde.
secondary, apart from secondary alcohol, there is also a primary
Primary
alcohol,
alcohol
because
group.
the concentrated nitric acid is a powerful oxidizing agent.
Oxidizing agent.
And HCN, reaction with HCN and hydroxylamine helps us identify the presence of proof for the presence of aldehyde.
Aldehyde group.
Secondary alcohol. Secondary alcohol. secondary alcohol. And the fact that the secondary alcohols are present on 4 different carbon atoms.
Yes, ma'am.
5 OH groups, out of which 4 are secondary alcohols and they are present in 4 different carbon atoms. The formation of stable pentacetate with acetic anhydride. Correct. When we have that formation of stable pentahydrate with acetic anhydride, they're stable because they are bonded to 4 different carbon atoms. In fact,
Different
when we include the primary, it becomes 5 pentacetate. We say that's why we call it pentacetate, because we include the primary alcohol also with that. So when it's stable, that's possible only if each carbon contains only 1 OH. So after doing this, giving them an idea about how the structures evolve, there's a lot of history behind it. We know it, but we don't give that to children. From glyceraldehyde, we start and we keep increasing the carbon, and then we come up with a structure of D-glucose. And one way I encourage children to remember is one structure and understand the difference. Remember one structure. If it's learning alpha and beta, remember what alpha is and understand the difference between alpha
And then
and
beta
beta.
difference.
Yeah, so that they can regenerate it themselves. That's one of the techniques to help them remember. So how do, please share your experience. How do you draw the structure on the board, or you put up a chart? How do you translate the structure to children? I'm looking for some insight here.
I'll make them to read only in the right hand side of the carbon. If the right hand side hydrogen is there, left hand side they can put OH like that.
Okay, only one
plan.
side you ask them to learn thoroughly.
Yes, yes.
So that one where there is
I can
hydrogen-
share my own idea, ma'am.
Yes, sir.
First I want to write. I will write the aldehyde group, ma'am,
Okay.
because it is a bifunctional molecule. More number of hydroxyl groups are present. Only one aldehyde group is present, ma'am. That's why I write the aldehyde group at the top. I give number 1 for that. After that, I write 5 carbon atoms. Next chain, ma'am. Next, I put hydrogen and every carbon atom except the 6th carbon contain one hydrogen atom and one hydroxyl group, ma'am. That's why at first left side, at the 2nd carbon atom, I write the hydrogen atom, OH at the right hand side, ma'am. After that, for the 3rd carbon atom, I write oppositely left side OH group, right side hydrogen, ma'am. After that, for 4th and 5th carbon atom, I write 2nd carbon atom, ma'am. Left side hydrogen atom, right side OH group.
Correct, sir. Correct.
For the last carbon atom, I put primary
OH.
hydroxyl group, ma'am. That's why CH2OH.
Okay. So we have our own ways. What is comfortable to us, we either start. It's better we start from C1 carbon, that is the aldehyde carbon, and then move on, rather than randomly. And when we write the structure on the board, children see how we write. They
Yes.
follow the exact technique. So both ways I really appreciate, and I understand that it has its benefits. And thank you for sharing it. We could also try it. I normally go
Thank you.
opposite. I say like the 5th and 4th carbon OH on the right hand side, and I say ulta, opposite and opposite. So OH on the right hand side, from the 5th and 4th carbon, OH on the right hand side, opposite. So I ask them to learn like that. So they fix up the OH on the right hand side for the 5th and the 4th carbon, and then they say opposite. So it becomes OH and opposite. They get the D-glucose. Again, like I said, we have our own ways. That is what is important. Like what works will depend on the batch. In
Yes.
some situations, we don't have to do all this at all. By just one look at it, by just getting an idea of how this structure is visualized, they draw it themselves. I've had students who are whom I call future achievers. They come up with this structure so well that they volunteer to draw it on the board, because every time I start a class, I start with what was done, especially in biomolecules. I start with what was done the previous day. So there I ask children whether they would like to take up the opportunity to draw the structure. They come voluntarily. I don't have to pick up students at all. They just come. So they like that sense of achievement when they draw a structure on the board. So if we could also encourage them that way, to make an attempt, they start liking the subject. But unfortunately, we do this chapter quite at the fag end of the academic session.
Yes.
So that was about glucose and details of how we teach them to draw the pyranose structure also, because that is related directly to the cyclic structure, which is in turn related to the open chain structure. Going on to the structure of sucrose and maltose, we have quite a lot, so I'm going a little fast here. This is one way. What you see on the left hand side is one way. This is not flipped directly. The alpha 1,2 glycosidic linkage of C1 C2 carbon, C1 of glucose and C2 of fructose, writing one below the other. In our textbook, they would have given adjacent. They would have flipped the fructose and given it adjacent. This is an easier way they can remember. Though the maltose is C1 C4, so that shouldn't be a problem at all. Alpha D glucose. Structure of lactose. These structures are never asked. But nowhere in the textbook it says that it will not be asked. Is there any content anywhere they have mentioned that structures will not be asked? They have not said so, but it's an unwritten rule. Like we all believe and hope that the structure will not be asked, but then we cannot leave children as such. We need to prepare them. So we need to teach them how to learn. I make it a point to teach them how to write the structures, not learn by heart at all. Start from glucose, understand the difference every time. Fructose also. Fructose, there's no structural elucidation. Directly we have the structure of fructose, cyclic, open chain, and fructofuranose. So that's it's not included here, but then we have to teach that. We have to make it a point. When I'm redoing the PPTs, I'll include that. Then structure of starch, again, structure only. In the textbook, they have not detailed much about starch. Amylose and amylopectin part, 20-80%. Water soluble, water insoluble part. Always asked. Amylose and amylopectin. The C1 C4 linkage for the linear water soluble part. C1 C6 linkage for the amylopectin part. So glycogen basically and cellulose, the beta linkages. In starch, it's alpha linkages and glycosidic linkage. Again, one very favorite question is glycosidic linkage, peptide linkage, and phosphodiester linkages. What are the differences between them? When we say glycosidic linkage, I remember in one board exam, when the question definition of what is the glycosidic linkage was asked, child did not write any sentence. This monosaccharide, bond, oxygen, bond, monosaccharide. Do you think that answer deserves mark? Monosaccharide, bond, oxygen, bond, monosaccharide. That was the answer written for glycosidic linkage. What's your opinion? Do you think that should have been given mark, or what is do you think would have happened? Explain more glycosidic linkage was a question. Part of 3-mark question. Part of 3-mark question. Monosaccharide bond, oxygen bond, monosaccharide for glycosidic linkage. Do you think answer like that deserves mark?
No, ma'am.
What do you think should the child write, sir?
Actually, the glycosidic linkage, which is formed by removal of H2O molecule only, no, ma'am.
Correct, sir. They are not asking how glycosidic linkage is formed, sir. They are asking what is glycosidic linkage.
Okay, okay.
The monomers of carbohydrate connected by
Oxygen.
Connected by glycosidic linkage.
Connected through oxide linkage.
Oxide linkage.
Is glycosidic. That's what the child had written, no, sir. Monosaccharide, bond, oxygen, bond, monosaccharide. No sentences. Of course, it can be given, ma'am.
Can be given.
Can be given. We did give marks. Sometimes children express it like that. In fact, I remember one board exam, there was a question about electroplating. Am I right? Child had drawn electroplating. I think electro-refining. There was a question on it. Child drew the diagrammatic representation that they learned in 10th standard. Anode, cathode, the direction of current was correct. And it explained whatever question was asked. Mark was given. In chemistry, we give marks for equations, we give importance to structures. Why not for descriptions like this? Unless if it's a definition, statement. It's a definition statement, then we expect only statement. Is there any other input, Lakshmi ma'am, that you would like to add on to this?
No, ma'am. That's it, ma'am.
So,
They even draw structures and connect the two monosaccharides and indicate that this bond.
in yes, sir. Put it in the
Structurally,
square, yes. That part is the glycosidic linkage also fetches mark. So it does not necessarily say that it is formed by removal of water molecule. It does not say, it doesn't ask how a glycosidic linkage is formed. If it is asked like that, then yes, the child will have to mention. But sir, what is glycosidic linkage? So I think it should work. And that was amylose and amylopectin of starch, cellulose, not much details, only the structures given. Glycogen, in fact, they have not mentioned the structure also. They've just mentioned a line about it. So amino acids is the next part. Again, classification, I have not included acidic, basic, neutral. Here, only essential and non-essential amino acids. These are very typical. And amino acids are, we are talking about alpha amino acids only here. So we need to be very clear when we communicate that only alpha amino acids, and this bitter ion becomes very, very important here in this part. And apart from that, the second way of classifying acidic, basic, normally from the structure, all these are given in the tabular form in a textbook with an asterisk. So from the structure itself, we ask them to remember where we have one COH group extra, it becomes acidic. When the number of COH group and NH2 are equal, then it is a neutral amino acid. And when it has an extra NH2 group, it becomes, or any other extra nitrogen presence with a lone pair, it becomes a basic amino acid. So pick up an example. One technique is pick up an example which works both for both type of classification. That way they'll be able to remember less. Learning by heart will become less. So we can think of a method like that, where we can also bring in classification bases, acidic, basic, and neutral amino acids. Essential amino acids, non-essential, we are very good at this. We keep doing this, and we keep telling children about how to remember this. Then moving between amino acids and proteins, we have this peptide, peptides, dipeptide, polypeptides. Let's just get something clarified about dipeptide. Dipeptides are biomolecules with two amino acids. Am I right? Children generally misunderstand dipeptides to be two peptide linkages.
Only one peptide linkage
Only
by
one
the formation of two
amino
acids.
Yes.
And this we have to emphasize, because in the board exam, when they say explain the following and they ask dipeptide, they write two peptide linkages in a biomolecule is a dipeptide. No, many children have lost marks. Dipeptide is formed when two amino acids are linked by a NHCO linkages, a peptide linkages. So that from there we evolve from amino acids to polypeptides. Polypeptides, that is protein. The definition of protein, and very important apart from definition of protein is what is a native protein and what is denaturation of protein, though not mentioned here because I'm more interested to talk about the structure, layers of structures of protein, which is always a point of contention for many of us, feeling as to how to communicate this. So denaturation of protein is very, very important. Apart from the definition, we have the classification of protein as well as. Classification is as globular protein and fibrous protein. One example each would suffice. That would help them get an idea about the classification of protein, denaturation of protein, and what is native protein. Because denaturation of protein definition is when native protein is subjected to change in temperature, pH, it undergoes changes structurally, only its primary structure is retained, it loses its biological activity and all is mentioned. But then what is a native protein? A native protein is a protein which exists in the biological system. That's a native protein. And denaturation example is when egg is boiled. That's one very, another, can you think of another example that you could easily come up with in the class which they could understand where denaturation of protein takes place? Anybody?
Can we take the example of milk, ma'am?
Yes, sir.
Generally, the lactose is present in milk, but the protein also there, ma'am.
Yes,
And
sir.
sometimes the nature of milk is damaged by some reasons.
Yes, sir.
Some
That also is
envi-
curdling of
yes,
milk
ma'am.
is what you
Yes,
mean.
ma'am.
Yes,
Yes,
sir.
ma'am.
That naturally also can be done, and it can be done artificially also for various purposes. So
Yes,
children
ma'am.
relate to that very well, where protein is separated. Protein
Yes, ma'am.
undergoes a change. Correct, sir. Egg,
Yes,
boiling of egg also. This is
Yes,
this
ma'am.
question. What happens to the water in boiled when egg is boiled? It's a textbook question. What happens to the water when egg is boiled? What happens to the water part of egg, raw egg, when it is boiled? Denaturation takes place. So talking about native protein and denaturation is very important part of protein, and in that denaturation, loss of biological activity is very, very important. Loss of biological activity is very, very important. Anybody can throw light on this question which children normally ask, ma'am, then raw egg is more kind of useful if I'm looking at a protein rich food. Should I eat raw egg or should I eat boiled egg? This is a very favorite question when we say when you subject protein to a temperature change, it loses its biological activity. So protein undergoes some change. So will it be useful for me if I boil it and eat it, because it's that way manageable to eat when compared to raw egg? What would be your answer if I am the student? I'm asking, if I'm denaturating egg, I'm subjecting egg to denaturation, and I'm by boiling it and making it hard boiled egg. Will I get the protein as it is, or is it undergoing a change, a biological change?
Generally, that before the boiling, the only microbes can there, ma'am. But after boiling, we can get the very clean food, ma'am. That's why we can
So
eat
that's why
only.
we encourage people to, especially small children, are given only boiled egg. But
Yes,
the
ma'am.
question is, will subject to change in temperature, egg lose its biological activity? It's a textbook question, sir. I don't remember which page, but there is a question on what happens to egg on boiling.
Coagulation.
Coagulation, ma'am. Prabhakaran sir, your thoughts on this? The child asks, is it beneficial for me to take egg? Of course, we say when you boil egg, it's more clean. Salmonella is there even in boiled egg. That's another issue. But then
Proteins will be lost only at high temperatures, ma'am.
proteins will
That
be
we
lost
can
only
see.
at high temperature. So
shall
boiled
we
egg can
give
be consumed. It will have that exact amount of protein, or the protein supplement will be provided as a raw egg, ma'am. No, we definitely will get questions like that. I don't know about all of you all, but I have got questions like this. In fact, I ask them when I get 10 counter questions like this, I don't give an answer immediately. I tell them, please wait, I'll get back to you. Because it's better to research on it and talk about
Yes,
it rather
yes.
than give them an answer which is convenient for us. They will not be convinced. They are 16 years old and
Sure,
17 years
sure,
old.
ma'am.
They'll immediately find out, okay, ma'am is not very sure. Yes, Lakshmi ma'am.
Normally what you said no, that we don't have lighter moments in chemistry. Normally when I introduce protein, I would say that all living beings have got 20 amino acids. Buffalo, a tiger, or a human being, or a rat, or a cat, all are made up of these 20 amino acids, maybe in a different arrangement. The sequence is different. So the answer to your question will be like when we boil the egg, the protein gets denatured, its primary, secondary, and tertiary structure that linkages are broken up. So it becomes all the more easier for a human being to break it down further, absorb it as amino acid, and resynthesize what protein we require.
Okay. So when the complex structure of protein is broken down, it becomes
So
easy
what
for
we-
the protein.
Yeah, normally the protein present in a hen which is laying the egg may not be useful for our body directly. So what our body does is it takes the nutrient, breaks it down, resynthesizes according to the body need, and then uses it. So when you are denaturing, you are actually breaking down some of the original linkages so that it becomes easy for our body to assimilate.
So am I to understand that the biological activity is not lost on boiling of egg?
Biological activity of the protein is lost, but what we need is not that protein, but we need the
We
protein
don't need the biological activity of that
Of that,
protein.
yeah.
We need the protein.
protein. So we need actually the raw material which made up the protein. We need the amino acids.
We need the amino acids
acids.
and that makes the protein, and we can
Yeah.
resynthesize after we use that protein.
Yeah.
So
I would normally say this, and they will be finding it very funny that a buffalo, a tiger, a hen, or a cat, or a human being, all of us are going to be using the same amino acids, but in a different order.
synthesizes in a different way to generate the proteins. Correct. So, like, yes, this is one chapter we can talk a lot. We can give a lot of inputs when we do a proper research on it. Going to a class and trying to face a class and trying to give them some questions for which we are not very sure of is a very bad idea. So again, this comes out of more and more students interaction. Many a time we prefer it comfortable because we have a target. Our target is to finish the portion in 1 week's time, 3 days time, 4 days. We have a target. So we make sure children don't talk much. And when they talk, also said you ask the doubt later. But then
Yes.
when we do that, when we say you ask the doubt later, the child will never come back. Even if he comes back, the doubt we might be able to answer to him. But then the bigger crowd, which probably had the doubt and never asked, those children will miss out on it. And from that answer, there are other doubts that will come up. A class which is very interactive with lots of doubt is a very successful class. The child go back. The takeaways are more when the interaction is more in the class. Like we had when we, so the fourth day today that the interaction has increased. And when the interaction increases, there's a lot of takeaway. I took a lot of things today from what Sir Manvati ma'am said and what Kripakaran sir said, and the other gentleman also said about how we remember the structure of glucose. So when we have our own ways to deal with things, and that is what makes these sessions very important. Mila ma'am also was saying about how CS children, we all understand that they need to be given a special way of handling this particular chapter. So talking about this is something we already know. Alpha helix, which is generally formed as a secondary structure, beta pleated is not formed here. Again, visualization helps here. We understand that only the quaternary structure, the complex structure exists. The primary, secondary are just the layers of it. And primary structure is often asked, what is the primary structure of glucose? Is always every time a part of biomolecules question paper. What is the primary structure of glucose? And we already know what this is. The helical structure and the folding of the secondary structure gives us the tertiary structure, and the spatial arrangement of the tertiary structure gives us the quaternary structures. So primary, secondary, tertiary, and quaternary structures. Primary is the sequence of amino acids. Secondary is the shape of the polypeptides, the sequence of amino acids. Shape of primary structure gives us secondary structure. The folding of the secondary structure gives us the tertiary structure. The spatial arrangement of the tertiary structure gives us the quaternary structure of proteins. So it's the quaternary structure which children kind of find it difficult to visualize. At this point, slides like this, or if taking them to the CS lab, or an audio-visual lab could really help in this chapter. Enzymes, ma'am, enzymes there, ma'am. It's there in surface chemistry. I'm not going to go much into enzymes. It's only oxidoreductase enzyme is mentioned because it's probably handled already. Surface chemistry is evaluated, ma'am, this year.
It's deleted, ma'am.
Deleted.
Chemistry. So that
Welcome back, Sindhu, ma'am.
Yeah, thank you, ma'am.
Your task is over. Important task.
I joined by 5, ma'am. That's what missed out.
No, not at all. We are talking. Today's session was a little more interactive than I got to learn a lot. So probably that part you missed. Otherwise, it was it's going on the same way. I
Ma'am,
hope you have
just,
taken down the attendance, ma'am.
Yes, ma'am. We have noted. Yes,
Yes,
yes.
ma'am. Yes,
Yes.
Lakshmi ma'am.
Ma'am, just checked on the curriculum. It says hormones, elementary idea, excluding structures.
Okay, it's there.
It's there, yeah.
There. Thank you, ma'am. Thank you for the prompt input.
Yeah, because we always need to check. Hormone is always there in the end of that book. So we always have a doubt and we always keep checking. So I thought I'll give it a check now.
Yes, ma'am. And hormones is another topic in biomolecules. So much in biomolecules, and focus is only on carbohydrates. Lot of dilution, small bits, and then proteins. And as we go down, it becomes lesser and lesser. And suddenly, nucleic acids, there's a lot. So this is not fairly distributed at all. What to learn and what not to learn. Definitely in enzymes, biocatalysts, the definitions, and the lock and key model, the complementary shapes. This comes very useful when this part becomes very useful when we talk about the enzyme in chemistry in everyday life. When we talk about the drug action, the action
Yes.
of chemicals as drugs
Active
in,
site.
yes, active site. So that there it comes as handy. So this is something which I'm not going to touch much upon because we already have done this. This hormone secreted by ductless gland, and its physiological, its activity is in place other than where it is secreted. Very, very important. Apart from the fact that it is carried to the bloodstream. I think these are the basic hormones. Their classification also is not mentioned in our textbook. Non-steroids or steroids, just the functions. Going through it's if my memory is right, it's just 1.5 columns mentioned in our textbook. Only that much is mentioned. You can give them a tabular column like this. What is the chemical name? What is the hormone? And what is its function? Keep them handy, especially for CS students. Biology students will not find it difficult at all, but CS students might need help here. EG or CS students or any other elective other than biology, if they are taken, they might need help here. So these are some things which we already know, but how to put it to them in the form of a kind of written tabular column would help better. Vitamins also, this is picked up from the textbook. This is a good memory exercise, like start from vitamin A. What are vitamins? Definition itself by many people, required in small quantities, is the highlight of the definition. Am I correct? Required in small quantities is a very integral part of the definition, required for maintenance and growth. And classification based on solubility, was water soluble and fat soluble vitamins. And the chemical names, have they asked any time chemical names like vitamin C, ascorbic acid?
No, I don't think so.
Well, I don't think so, but then they have mentioned in the textbook. This is a textbook.
So anytime it can come.
It can be asked. So it's better we empower them with that knowledge as they know. Again, we have to tell them. We cannot say it's non-evaluative, don't study. That is a very bad idea. NCERT, whatever is there in NCERT, child should be thorough with every part of it. Again, these are the nine vitamins which are described in our textbook. It comes in 2 columns, am I right? This is put up in 1 column itself. I normally ask them to kind of chorus also. Ask them from A, B1, B2, vitamin B6, 12, C, D, E, K. And then what are the sources? 1 sources. First 4 milk, common. And the only source of vitamin B12 for vegetarians is curd. And of late children have started telling me rainwater also has vitamin B12. So they come up with various other sources of vitamin B12, at least in the textbook, what is given. But then there are other sources of vitamin B12. And this as a drill project also can help. What are the vitamins they have in their home every day, if they can list out and bring. So deficiency of what would cause what deficiency disease could be a very important drill project, a methodology where we can ask them to investigate as to what are the vitamins which they have taken in a particular day. Most of the days we'll find that we are going without any vitamin supplement in our food, dietary consumption. Am I right? I don't think all of us take all these every day. So asking a child to maintain a tabular column, a chart of what he has taken through the week and how much vitamins he is missing out could be a drill project. One of the methodologies to kind of very useful one also, because once he starts maintaining a record of his food intake, he or she will become more conscious of to make sure that they include food which includes these vitamins. Going on further, this is the last part. We just have time for some discussion after this. Nucleic acids, again, structures, we say the ribose made of the phosphate group and the bases, the difference between the bases. Generally, we give this rough identification of the first carbon where we have a nitrogen base, the fifth carbon where we have a phosphate group, the fourth carbon where we have the phosphate group, no, to the fifth carbon only, I'm sorry, we have the phosphate group, and the difference between ribose and deoxyribose. This is again a little challenge because a lot of complex structures given, the structures of adenine, guanine, thymine, cytosine. This is something which children keep asking about whether they should learn the structure. In fact, I remember one year in board exam, they just said, what are the bases present in DNA? Just the caps of the first letter of the base is enough. I don't know. A, they write A, G, T, they are getting marks. But then it's a bad idea. It was only for children who had needed that marks to get that 33%, only we gave that marks. So please encourage children to write the complete names. Spelling errors doesn't matter because they are worried about how they're going to spell guanine, how they're going to spell adenine, cytosine, and thymine. It's okay, but then let them write the full, not just say A, T, G, C. Doesn't work like that. Structures, do you encourage children to learn structures in bases, nitrogenous bases? I don't.
No.
We don't do it. Should we do it?
There is
I
always
don't do
a
it.
note, no, that the complicated
Linkage.
structures in
We just need to know, ma'am, which linkage, base, sugar, 1, 5, that sort. Structure is not needed at all, I think.
But then nowhere it is said, ma'am, structure will not be asked.
Not asked, yes.
But what I always add is like this is one content which is probably the last choice of the examiner. And if you still want to make sure that you don't want to miss any marks, maybe if you have energy,
Yes.
you learn. That's
Yeah,
how
if you have the time and you have the patience, do learn, because there's so much more that is there in chemistry, part 1 and part 2, first part. This you have never commit and say this will
Tell
not be
that
asked.
never has been, it's not been asked, that's all, we are not sure. That's all we do. Yeah, we cannot
better
say
thing
not to
say.
learn.
Yeah, better is that safe thing to that. If the child has the capacity, I knew some children who used to know those structures by heart. It could come handy for them for their competitive exams. You never know. So children, we think that they will not be able to remember, but they do. Some of them make it a point to remember and they enjoy learning it also. So why deny that pleasure from children and say that this will not be asked in the exam? Don't learn it. Don't ever say that. I never do that, but I don't emphasize on at least the structure of the basis, learning by heart. So again, the double helix structure and the single helix, the difference, normally RNA and DNA, what are the difference starting from the base, from the sugar, from the base, the function wise is what is compared. Not much details given in chemistry textbook, though in biology, I understand they have more details of this. The one very famous question is the strands in the double strands in DNA are always complementary, never identical. This is a very famous question. Again, we know the answer. How do we put it across? Again, like best way is visualization. Most of the laboratories have the double helix model. Yes, we can procure one. It could be useful biology. They have a chart, at least leaves they have is a chart. If not the model, but then model also with ball and stick, it is easy to prepare a model. It will take a lot of time. But if we have the right kind of balls and sticks, we can prepare a double helix. And that could be a project as well to help them, really get the feel of biology and chemistry together. As to what do we say about the strands being complementary and not identical? Because of complementary base pairing, GC bonds only with cytosine. We say about complementary base pairing, AT, adenine with thymine. So that's where the strand, the base, the nitrogenous base in one strand decides the sequence of nitrogenous base in the second strand. So they are never identical. They are always complementary. We talk about the hydrogen bonds, the number of hydrogen bonds, and we talk about the types of RNA. And RNA has become a spotlight after COVID. What is the necessary small inputs, like why is it necessary to wash hands, and what medicines could not do, a soap water can do. That doesn't mean you drink the soap water. There's lots of funny forwards about how a soap water can prevent COVID. A little input about what is about the RNA and what is the role of soap water. Could really give at this point, the COVID time can help them understand it a little better.